In this video, we'll be trying to understand the process of Megasporogenesis, that is the development of the female gametophyte or the embryo sac. So here we are going to just understand the process by which the embryo sac formation is done or it takes place. Let us come back to the structure of the Anatropus ovule that we have studied so far, that it consists of the funicle which attaches the ovule to the placenta. Then it has the integuments, that is the outer integument as well as the inner integument. And here this we are just labeling it first. This is the funicle over here. Then this ovule has two ends. One is known as the chaliceal end and the other is known as the micropylar end. These are the two integuments, that is the inner integument and this is the outer integument. Coming to the inside, what we are focusing over here is the cells which are known as nucellus, which is known as the nucellus. Now this nucellus is arranged compactly over here. The cells over here are arranged compactly, and out of these cells which are present over here, only one cell which is towards the micropylar end. It is very important always to label the chaliceal end as the micropylar end, because it is through this micropylar end the pollen tube will enter during the process of the transfer of gametes. So here the nucellus, which is densely packed and with the diploid cells, it is from here that only one cell will develop, and this cell will grow in size and is known as the megaspore mother cell. Megaspore mother cell or MMC. Now this cell is present towards the micropylar end. Let us take and study this cell, how and what happens to this cell. This cell grows bigger in size, that is the MMC, and it is actually a diploid cell. This MMC will undergo one meiotic division. It will undergo one meiotic division, and as a result it will form a tetrad. The tetrad in case of angiosperm is always linear. It is always a linear tetrad. Now this tetrad which is being formed over here, it is due to the meiotic division, only one meiotic division, it will form 4 haploid cells. And always remember, as we are doing the labeling part, it is important to mark the micropylar end and the chaliceal end. So this is the chaliceal end over here and this is the micropylar end. Now this linear tetrad is formed always in the case of angiosperms, because we are studying about the Anatropus ovule, which is about 80% of the angiosperms have this type of ovule. This linear tetrad which is being formed over here out of these 4 cells which are now haploid, it is now having single set of chromosome. Here the MMC is diploid. Now out of the 4 cells, the 3 cells towards the chaliceal end will degenerate. So if we just make it this way, the 3 cells towards the chaliceal end will degenerate. and out of the 4 cells, only one cell which is towards the micropylar end, that is this cell, will now form the embryo sac. So it is known as the functional megaspore mother cell. It is the functional megaspore now. This is functional megaspore, and this functional megaspore is haploid in nature. And since the embryo sac, that is the female gametophyte, develops from one megaspore, the condition is known as monosporic. We also call it as a monosporic development of the female gametophyte, because out of the 4 cells which are being formed from the MMC, that is the megaspore mother cell, 3 degenerate towards the, which are to present towards the chalazal end, whereas 1 which is present towards the micropylar end continues to survive, and it is now known as the functional megaspore or monos. That is why we call the development as monosporic. Now let us understand how this megaspore forms the embryo sac. This megaspore, that is functional megaspore, it undergoes 3 repeated division. So it divides 3 times, and remember, all the 3 times this division is mitotic. 3 times it divides, and these divisions are mitotic in nature. That means the chromosome number, since already it is haploid, the chromosome number will remain the same. Now, as it undergoes 3 mitotic division, also important for you to understand is all the divisions are without cytokinesis. That means karyokinesis is taking place. Karyokinesis is taking place, but cytokinesis, that is the formation of the cell wall, does not take place. As a result, it will grow bigger in size, and it will form nucleuses, and these, let us try and understand. As a result, the first time the cell divides into 2 due to the first mitotic division. Due to the second mitotic division, it will form 2 cells, and due to the third mitotic division, it will form 8 cells. But remember, as told, the cytokinesis, or that is the cell wall formation, is not taking place over here, but this will have 8 nucleus. Now, out of these 8 nucleus which are being formed, and these are all haploid, the mitotic division has already taken place, the meiotic division has already taken place, which has reduced the diploid condition to haploid, and out of this one cell, which is functional megaspore, will undergo 3 mitotic division to form 8 nucleus. But these 8 nucleus are haploid in nature, and no cytokinesis has taken place. Later on, out of these 8 nucleus, again we are marking the chalazal end and the micropylar end, chalazal end and the micropylar end again over here. Out of these, 3 will move towards the chalazal end, whereas the 3 again from this end will move towards the micropylar end. Now let us see the arrangement now. So this is the embryo sac which is being formed over here. Now, as a result of moving of the 3 nucleus towards the chalazal end, we have 3 nucleus over here, 3 nucleus over here, and 2 nucleus which are present over here. After the movement of the nucleus towards the chalazal end and the micropylar end, the cytokinesis takes place. Now here, the process of cytokinesis will occur. That means the formation of the cell wall. And as a result of the formation of the cell wall, the cells which are present over here will develop the cell wall. So let us draw the cell walls around these. These three cells which are towards the chaliceal side, and the three cells which are present towards the micropylar side. They develop the cell wall. The two cells are present over here. Now the cells which are present towards the chaliceal end are known as antipodal cells. That means the three antipodal cells are present towards the chaliceal side. Towards the micropylar side when we come, there are two cells and one middle cell which is slightly longer. So this cell on either side are known as synergids. So we have two synergids and one middle which is appearing big one is actually the egg cell. So we have one egg cell and together this is known as egg apparatus. Remember, the egg cell is present towards the micropylar end, and micropyle is the side where the pollen tube will enter for fertilization after the pollination. Now these two nucleus which are present in the center are known as polar nuclei. So there are two polar nuclei. If we talk about the embryo sac, this is the structure of the embryo sac. Now if we study or just go through it again, this is the Anatropus ovule which is very commonly found in the angiosperm. More than 80% of the angiosperms have Anatropus ovule. Now these are the integuments, and inside is a mass of cells which is known as nucellus. One cell out of the entire cell of nucellus which is present towards the micropylar end will grow bigger in size and it will divide, which is since it grows bigger in size and will undergo further division, it will be having dense cytoplasm and it is known as Megaspore mother cell or MMC. This Megaspore mother cell undergoes one meiotic division to form a linear tetrad. That means in the angiosperms, the cells after one meiotic division will be arranged in a linear pattern from chaliceal end to the micropylar end. And out of this, the three cells which are towards the chaliceal end will degenerate, leaving only one cell, and that cell will be known as functional megaspore. Now since the MMC has undergone meiotic division, it will form haploid cells. So these functional megaspore mother cell is a haploid structure, and since the entire embryo sac develops from one cell, that is why it is known as monosporic. This functional megaspore undergoes three mitotic divisions, which is characterized by having karyokinesis only but no cytokinesis take place, and due to which there is a formation of the 8 nucleus. And since no karyokinesis has taken place, that means we also call these divisions as free nuclear. No cell wall formation has taken place till now. So as a result, we have chaliceal end, micropylar end and 8 nucleus. Now, later on, these 3 nucleus move towards the chaliceal end, whereas another 3 move towards the micropylar end. Haploid is the ploidy of these cells which are nucleus which are present within the embryo sac. Once they have moved, the formation of the cell wall takes place, that is the cytokinesis. And as a result, we have 3 antipodal cells, 2 polar nuclei and 3 cells, out of which one is forming the egg cell and 2 synergids. The egg apparatus is the term which is given commonly for them. Now this egg apparatus lies towards the micropylar end. Now, if you see carefully, the number of nucleus, let us count the number of nucleus in the case of embryo sac of Anatropus ovule. There are 3 antipodal cells, 2 polar nuclei and 3 cells that comprises 1 egg cell and 2 synergids. So, here we have 8 nucleus, or we call it as 8 nucleate. 8 nucleus over here, that is 8 nucleate. Coming to the number of cells, when we talk, this consists of how many cells? The 3 antipodal cells that is present over here. Due to the formation of the cell wall, we are calling it as cells. Here we are counting the nucleus only. So 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleate. We write 8 nucleate. Cells we are counting. So 1, 2, 3, 4, 5, 6. And remember, the polar nuclei has not developed a cell wall around itself. So they will be fusing later on to form the endosperm. But here we are counting the cells which have formed the cell wall. So 1, 2, 3, 4, 5, 6. And this is the 7. This large is the 7. So it is 7-celled. So if we say the Anatropus ovule of an angiosperm is 8 nucleated and 7-cell structure. This condition is known as polygonous condition. This situation, when it is 8 nucleate and 7 cell, it is known as polyovule or condition, we call it. Polygonous type of ovule. In the case polygonous condition. So here the angiosperm ovule, if we ask what is meant by polygonous condition, that means it is 8 nucleated but 7-cell structure, and it is monosporic because of the reason that it is formed from 1 megaspore. So here this embryo sac is present then within the ovule, and which will be later, this egg cell which is present at the center will be later on fertilized by the entry of the pollen tube which carries the male gametes.